Hello, are we live? Can you guys hear me okay? Give me a thumbs up or something if you can see and hear me. Perfect. Sorry, I don't know why my video is so blurry. I was trying to, the webcam's like not focusing. I was trying to get to focus for a second, and then it might be better now. Then it was like unfocusing. So welcome to this web class this evening, this GCSE web class. Just a quick show of hands. Who's joining us for the first time today, and who is a returning student? What's the mix? Zara returning, Fizzy James returning. Yes, I remember that name. You're still first time, welcome. Quite a few more returners. Hi, Amy. Hi, Ray. Oh yeah, Amy is your sister, and you're using her account. That's what it was. First time, Pfizer, welcome, welcome, welcome. Ultimate Revision. Sounds like a rival company to Snap Revise. Welcome to the class. Did you guys see the sad news today? Captain Tom Moore has died of coronavirus. Very sad. For those you don't know him, he's the one who did 100 laps of the garden on his 100th birthday and raised, it was like £33 million charity. It was not charity, for the NHS. So yeah, pretty special guy, but yeah, unfortunately passed away today. Nice. So let's make a start on the lesson today. So we looked at cells last week, and we looked at the difference between plant and animal cells. Today we're going to sort of expand from that. I'm going to look at prokaryotes and the eukaryotes. Mariam, this would be suitable for either triple or double. It relates to both. Zane, you do economics, maths, and psychology A-level. Yeah, I don't know if you'll get that much out of this today, but please feel free to tune in. Yeah, it's more geared towards GCSE biology. So hopefully you guys can now see a PowerPoint. Yeah, looks like it's working. Ramin, good revision. Are you A-level or GCSE? I'd say if you're doing GCSE, then it's definitely good revision. A-level, you might find it a little bit simple. Yeah, give it a go. The mic quality, I can try turning up a little bit, but usually, is it quiet for everybody? Maybe the cable's loose or something. Is it any better? Okay, perfect. Yeah, I just turned it up a little bit. Sometimes, yeah, it's difficult. Sometimes if it's too loud, then it picks up every single thing. Like it picks up my breathing, which is very distracting for everyone. So it's muffled. Strange. Actually, that's not the case. Let me just check it is actually. I think it is the right one. Sorry, in answer to your questions, GCSEs, I wouldn't say they're more important than A-levels. Other than GCSE maths and English are very important because you still need to have a GCSE in maths and English for basically any job. It's like one of the things I'll ask you, do you have a pass grade in English and maths GCSE? So those are very important. If you then go on to do A-levels or another qualification that supersedes them, then they're of lesser importance. But yeah, they're going to be on your record for the rest of your life. So yeah, they are important, definitely. Okay, but yeah, so say if you didn't do so well in your GCSEs, if you then got a very good A-level results, that's gonna be like look good. It's gonna show improvement. The only thing is your application to university, you're gonna send that off without any A-level grades. So they're gonna base that on your predicted grades. But because predicted grades, there's a certain amount of people can say whatever they want. They do also look at your GCSE when they're deciding for university courses. So if you want to go to particularly like a prestigious university, they are going to look at your GCSEs definitely. So yeah, it's important for that. Okay, so a little bit about me. My name's Alex. I'm the head of biology here at SnapRevise. I've been teaching and tutoring biology for the last 4 or 5 years now. SnapRevise is a revision website. It's been running for a few years now for A-level content, and we're now moving into GCSE content as well. So that's why we're running these YouTube classes, kind of as a little taster of what we're going to be offering on our website. So for now, we're running them on YouTube, but after when we start up this month, we're starting. It's going to be a subscription-based package on our website. So hopefully, we'll see all of you guys signing up for that. So yeah, what we offer, we're going to do four or 5 classes a week for each subject, going through all the curriculums, all the syllabuses. Doesn't matter which exam board you do, it's going to be relevant. Going to give you like top quality cheat sheets, crib sheets. So this is like really condensed notes for you to revise off. They're really helpful. And all of the past recordings will be available to download. So if you miss a session or don't want to go one evening, you can catch up on it at any in the future. Joining any university is going to be like wanting to see pass grades at GCSE, really, and then top universities are going to want to see like good passes. So yeah, there's no like set thing, but the better you get, the better it looks. But I mean, yeah, the practicals is a good idea actually. I was thinking about this the other day about doing that for A-levels. I think that would be actually a really useful thing. So maybe that's something we're just looking for new ideas, and we're very open to feedback. So if there's something you guys want to do, quite often we can implement that and bring it in. So yeah, that's definitely a good idea. Okay, so a little bit of excitement. We are giving away 10 free accounts in celebration of the new course. So to begin with a chance of winning, you just need to go to this website and enter your email address. And that competition ends on the 15th of February. So we'll draw the winners on the 15th. So might as well enter, nothing to lose, and potential to win an account. So all right, yeah, we might stop. We might get down to like doing 1 a month. That's what we do with A-level. I'm not exactly sure what we're going to do for GCSEs. But yeah, these are kind of like a taster to show you what will be offered on the website, and hopefully to entice you guys to sign up and at least give it a go, maybe for a month, see how it goes. How will you find out? I presume they'll email the winners. Again, I'm not exactly sure how they're going to do the format, but yeah, if you win, you'll definitely know about it. Johnny, I don't know. I don't want to put like numbers on what Oxbridge applications are going to be accepting. It's going to be on a case-by-case basis. So someone with extenuating circumstances who kind of bombs on their GCSEs isn't like definitely not going to get into Oxbridge, but in general, they want 7s, 8s, 9s for your GCSEs, and that kind of it would be unusual for them to accept people with like significantly lower grades than that, because they don't need to. There's so much competition. They can choose to sort of take the higher grades. Okay, so the objectives today, we're going to look at the difference between eukaryotic cells and prokaryotic cells. We're going to explain how the structures inside prokaryotic cells relate to their function, so a bit like last week, but with prokaryotic cells. And then we're also going to look a little bit at chromosomes. So let's dive in. These are the spec points. I'm just going to flick through them quickly today, so we've got more teaching time. But if you want to screenshot your spec, then please feel free. Edit cells. You can obviously get these online as well, the specifications. And I recommend that you guys should all print out a copy of your relevant specification, just so you can refer to it whenever you're like, oh, do I need to know this bit, but not this bit? Just quickly refer to your syllabus. Really handy. Make sure you're not learning things you don't need to know. It's not disaster. It's always good to have additional knowledge, but it's a real challenge just to learn all the content in biology. It's a massive syllabus, so I wouldn't like prioritize things you don't actually know. Yeah, Johnny. I do, and there's a bunch of other tutors as well. So I think we now have two for each subject, maybe even a few more for some subjects. So yeah. Okay, so what do we already know about cells? So some of you were in the class last week, you'll be able to tell me, or you guys know anyway. So what are the similarities and differences between plant and animal cells? So similarities and differences. Over here. And over here. Both have DNA and a nucleus. Nice. Yeah, like that. Both have mitochondria. Let's just list all the organelles. Ribosomes. Cytoplasm. Membrane. So, both are eukaryotic. Nice, yeah. EUs, I've run out of room. Cool. How about some differences then? Anyone put any differences so far? I think would be ultimate revision. Yeah, nice. So they're differences. So cell wall in plants. Cell wall plus vacuole plus plant only. Cytoplasm in plant cell only. No, they both have cytoplasm. Ramin, yes, that is very true. So we've got to be careful when we say vacuole. Animal cells can have vacuoles, indeed. So when we say like vacuoles only in plant cells, what we mean is large vacuoles. So only plant cells have large permanent vacuoles. So this is the vacuole, or the large permanent vacuole. But you are indeed correct. They can be in animal cells. Small ones. Okay, so that's, yeah, happy with that. Well, let's recap. What is DNA? How would you guys define that? My screen keeps like going black. Does it look black for you guys? Hopefully not. I think it's just like a wires a bit dodgy maybe. Yes, like in these answers, so made, or it stands for deoxyribonucleic acid. Yeah. And it contains, yeah, the genetic code of the organism. Nice. So it's found in the nucleus in eukaryotic cells at least, and contains all the genetic information, the instructions for making proteins, essentially. Yeah, made up of 4 different types of bases, A, T, G, and C. Nice. And yeah, you need to know the full names of them. We're not going to dwell too much on that today. But yeah, you definitely need to know the full words. Nice. So, moving on. Eukaryotic and prokaryotic cells. So let's just firstly recap what we mean by multicellular and unicellular life. So multicellular life, what are some examples of multicellular things? What do you put that in? Yeah. Multicellular organisms made up of more than one cell, so many cells, like I said by the boss. Yeah, elephant, like that, yeah, humans. Yeah, so animals. Nice. So what else we have? Small countries, birds, trees. Um, yeah, all this stuff is multicellular. We've got some examples of single cell things. Maybe this is a little bit harder. I'm going to add your leader because that was a good one. What else is single celled? Yeah, nice. How is it? E. coli, yeah, so it's a type of bacteria. Nice. And amoeba is a single-celled organism. I'm using the definition to explain it. It's basically like a little blob that can move around and engulf things and eat them. So they're very flexible. They can contort their bodies in lots of ways. Watch them under a microscope. Quite cool. Yeah, very simple. Like diatom. I like that one. I mean, yeah. So diatoms form the sort of start of pretty much all the food chains in the ocean. So they're photosynthetic type of algae, so they can convert energy and light into glucose, and then they get eaten by bigger types of algae. Sorry, not algae, bigger types of plankton called zooplankton, and then zooplankton get eaten by bigger organisms. The up the food chain in the ocean. Yeah, for those of them. Nice. And there's loads of different types of diatoms and plankton. Yes, Amy, you're right. This is a type of bacteria. So I could have done that as a line. Nice. Yeah, algae can be single-celled, rare, and it can also be multicellular. So seaweed is a type of algae. And you've seen videos like a kelp forest. That's obviously like multi-celled. You can also get single-celled. So yeah, those kelp forests they can be like the kelp strands themselves can be up to like 100 meters long, so they can go like really multi-cellular. You guys should watch. I'm talking well. You should watch My Octopus Teacher if you haven't watched it yet. It's good. It's all filmed in a kelp forest of South Africa on octopus. It's a nicely filmed documentary. It's on Netflix, I think. Oh, there's banging noises. That's probably because I turned the sound up too high then. Let me put in an earphone because then I can hear what's going on. I usually do. Yeah, that's why I was worried about turning it up too loud. Oh, I don't know why it's muffled. Let me have a listen, see what you guys are. Is that any better now? Yeah, slightly better. Okay, perfect. Oh, I wonder what if it's. Let me just quickly double check it is using the right microphone. Yeah, it's on the right one. So I'm not sure why the sound is not good. Okay. Nice. So, oh, and yeah, this is how an organism is made up, a multicellular organism. So you've got cells, many cells together make up a tissue, multiple tissues working together to form a function, call that an organ, and then multiple organs working together is an organ system, and then lots of organ systems make up an organism. Yeah, the drawing would have gone. I cleared it to check the microphone. Muffled. I don't know why. Yeah, I'm really sorry about that. I don't know why it's having problems. There's nothing. Everything's like plugged in. Can't really see any reason that it's not working very well. I've turned it down a bit so that it won't pick up other noises as easily. Okay, it's fine for some of you now. Okay, that's good. Okay, nice. Good. Glad to hear it. Oh, the thing I was saying to what some of you said didn't catch it. It's called My Octopus Teacher. Just yeah, quite a nice documentary about this guy finding an octopus. Okay, so how did we evolve into the life we have today? So we got complex multicellular life. Only after many, many millions and billions of years of evolution. So what do you think came before multicellular life? From fish, yeah. So all land animals evolved from fish, but fish were already multicellular, even further before that. Yeah, nice, Sarah. So that's the endosymbiosis theory that the free living can't talk, mitochondria and chloroplasts were free living prokaryotic cells that got enveloped. So originally it was like a symbiotic relationship, and then it became from now they're like yeah, protists. That's one of the earlier categories that evolved. So they're a bit of a weird group. They're a kingdom that doesn't fit in organisms. They've got some traits which resemble plants, some traits which resemble so organisms. They can be like photosynthesis, they can function in a saprophytic, so they resemble fungi. Lots of weird ones in that group. So yeah, we had so multicellular life are all the eukaryotes. We also get some single cell eukaryotes, but the majority of them are multicellular. So before that, we had prokaryotes. And they can be split into two branches. You've got bacteria and you've got archaea. So archaea on the surface resemble bacteria. They look quite a lot like bacteria, but they have some eukaryotic tendencies. So yeah, they have some features which resemble more a eukaryote. Sounds distorting again. Yeah, I'll wiggle the cables to know if that's going to do anything. Any better? Yeah, I don't know why it's like. Okay. I just, yeah, just got a message saying that the sound is bad. Go again. Tell you what, I'm going to try switching to the like this microphone in case it's just that one. Okay. How is the sound now? Is it any better? It fluctuates. This is better. Okay. This is like, that's using the laptop microphone, which probably isn't as good, but if that's fine for now, then we'll go with that. Let me know if it gets bad, and I will try switching again. Yeah, I don't know why it's not working. This is like a top of the range microphone, and it never usually has any problems. So yeah, sorry about that. Okay, so yeah, let's look at a prokaryote in more detail. So does anyone know what these names, prokaryotes and eukaryotes, what those names actually mean? We can kind of split them into pro and eu, and then karyota. Yeah, nice. So it means with nucleus. Exactly. So karyote, it comes from the Greek word for nut or kernel, which is like resembling the nucleus. So yeah, eukaryotes are with nucleus, and prokaryotes, pro meaning like before. So prokaryotes evolved first, and then cells more complex. Life is always getting more and more increasingly complex as it evolves. So one of the major things involved was membrane-bound organelles, one of them being the nucleus. Um, yeah. So if you're ever in doubt, because it's easy to get these two words muddled up, just remember that you are a eukaryote, and so are all other multicellular things. So you are eukaryotes, prokaryotes, thinking of bacteria. So they are like more simple, no nucleus, and no mitochondria or any membrane-bound organelles. What do you mean membrane-bound? Samuel, which one is the virus? Good question. Neither of these are viruses. Viruses aren't even at the level of development as prokaryotes. They're not even living things. So viruses technically aren't alive. So they're neither prokaryotic or eukaryotic. Yeah, nice guys. So enclosed within a membrane. So this includes things like mitochondria, uh, endoplasmic reticulum, what other things, chloroplasts. They don't have any of those membrane-bound organelles. They do have ribosomes. Ribosomes aren't in a membrane, so they have ribosomes, but they're smaller. So, yeah, they still have those. Nucleus. Yeah, no lysosomes either, because lysosomes have a membrane around them. So yeah, they wouldn't have lysosomes. Yeah, they have plasmids, and we'll get to those later. So close. They are. You don't actually need to know for GCSE, but you do need to know for A level. They have 70 S ribosomes in bacteria, prokaryotes, and you have 80 S ribosomes in eukaryotes. But yeah, you don't need to have that for GCSE. Viruses are parasitic. Yeah, yeah. You can get parasitic bacteria though, as well. But yeah, viruses, the reason they're not counted as living is because they're not able to replicate by themselves. They're just very, they're simple, but they're very effective, like little bits of code that are self-replicating, so long as they can get into cells to replicate for them. So instructions to make more viruses that get into a cell, inject the instructions into the cell. There's various methods that can do that. And then they sort of turn that cell into a factory, lots of copies of the virus. That's how they multiply. It's very effective, but they're not actually living. And yeah, it's obviously effective because a coronavirus is holding pretty much the whole world to ransom. It's just how effective it can be, even though they're not alive. They can cause a lot of disruption. Yes, human, nice. They have circular DNA, exactly, which I think we're going to cover on the next page. Yes, the one after this one. Okay, so we said all eukaryotes have a nucleus, and the genetic information is included in the nucleus. So it's formed into these sections or structures known as chromosomes. Made up of DNA. So who can tell me how many chromosomes we have as humans? Or does anybody know any other animal chromosomes? I don't know off the top of my head. I think I was going to guess number of fruit flies, but I'm now not so confident. Yes, exactly. 23 pairs or 46 chromosomes. The only one you ever need to talk about or know by heart is humans. But yeah, I else knew any other ones off the top of the head. Interestingly, you might think that the number of chromosomes is related to how complex the organism is. Kind of makes sense. The more chromosomes, the more instructions you can have for making different proteins, therefore the more complex. But it's actually not that simple. There are very basic organisms with large numbers of chromosomes, and there's quite complex organisms with not that many. So actually, as humans, we don't have that many chromosomes. 46 isn't a particularly large amount, considering how clever we are, or at least we think we are. Um, we don't have that many chromosomes. There's many plants which have more chromosomes than humans. If you just look some of these up, mice 20, chimpanzees 48, dogs 39. Dogs have an odd number. That is interesting. That makes it hard to divide. Yeast has 16. Yeah, so there's not that much correlation. Yeast and mice, massively different levels of development. Yeast are single-celled organisms. Mice are obviously complex multicellular organisms. Humans, it varies for different plants. So some plants have a lot more than that. Some plants have less. Down syndrome in humans, yeah. So what Down syndrome is a chromosomal disorder. It's an additional chromosome. So so we said we've got 23 pairs. So they look like this. This is a pair. 2, 3. We've got 23 of these. If you have Down syndrome, what you have is on your chromosome 21, rather than having 2, so we sort of skipped a few here. 21, you have a third chromosome. So we call that trisomy of chromosome 21. And that leads to all the symptoms, which I'm sure you guys are familiar with, for of someone with Down syndrome. So yeah, very simple. Like basically, the DNA was not separated correctly in one cell, ended up with an additional copy, which will lead to different like proteins being expressed at different rates. 256 in a hermit crab. Is that true? That's a lot. Yeah. Abraham, what is your question? Yes. Also, we've gone slightly off topic. Does it sound bad again? Yeah. Okay, it seems to have like very split. Some people have bad sound, some people have good sound. Maybe it's like YouTube having issues. If it works for some people, then it must be like a problem with like sharing it from YouTube, rather than like the actual recording. This is more like going into A-level stuff. You don't need to know for GCSE. M1, the fundamental difference is prokaryotes don't have nucleuses and eukaryotic cells do. So let's look at things that prokaryotes do have. So we said they do not have a nucleus. So the DNA is not within a nucleus. It's actually just stored as a big coil in the middle of the bacteria, in the middle of the prokaryotes. It's sort of like free floating in the cytoplasm. It's not coated by a membrane. It's just free floating, hanging out there in the cytoplasm. So it's a big long loop. If you straightened it out, if you uncoiled the coil, it would actually form a circle. So we say that their DNA is circular. So yeah, you can unravel it and it would form a circle. Whereas in eukaryotes, if you unraveled our DNA, it's linear. It would form straight lines. So that's another difference. So loose DNA, not in nucleus. Yeah. Still, some people having issues. Yeah, sorry, I don't know. My thinking is that it must be a problem with YouTube. If some people can hear it and some people can't, then it must be recording correctly. So I don't think there's anything I can do. Evan, they have 1,260. Wow, that is a lot. Yeah. Lots of banging. Yeah, let me try talking back to the other microphone then. I think it's actually not the microphone that's causing the problem. Which will be slightly less banging sounds. So yeah, so we call this circular DNA. Okay, good. Yeah, this is back to the proper microphone. So nice. Glad that's working better. We'll stick with this one for the rest of the lesson.
So plasmids are small loops of DNA. So let's put that down. So these can be used, they have genes on them. Bacteria are quite clever in terms of they can actually exchange genes between them. So one bacteria can give its genes to another bacteria. We call that horizontal gene transfer. So we, as eukaryotes, we can only give our genetic material to our children. We can only pass it vertically from generation to generation. But bacteria can exchange them with each other, which is a weird idea. It'd be weird if we could do that as humans. So imagine, like, I don't know, you saw someone, they had, I don't know, red hair, and you decided you liked the red hair and you wanted to have red hair yourself, and they could give you that gene for red hair, and then you'd be able to have red hair. That's basically what bacteria can do with these plasmids. They swap the genes with existing bacteria, not to their children. So yeah, really cool. It would be interesting if we could do that for humans. Well, actually, to be fair, we're sort of at the stage in biology where we are able to do this. So with genetic engineering, we can actually insert genes into adult humans. So yeah, and it's going to be a field that's like taking off over the next few years. So yeah, we can't do it like bacteria naturally, but we are able to do it in the lab, and it's going to be really interesting this next few decades to see what happens. There's going to be a lot of ethical questions we need to answer, because we can now do things that we didn't used to have to worry about because it wasn't a possibility. But now that we can engineer so much with our biology and our DNA, it's going to raise lots of ethical questions. But yeah, certainly it's an exciting time to be studying biology and to be learning about biology. Yeah, it can. There is obviously risks associated with this, and at the moment, you're not allowed to edit someone's DNA without their permission. So this is most effective if you edit the DNA while the person's in an embryonic stage, because you can edit all of the cells more easily. But you're not allowed to edit an embryo's DNA because they haven't given consent, and they can't give consent because they're an embryo. So it's kind of like a catch-22. Anyway, yeah, don't want to go on about that too much. Embryonic stem cells, yeah, that's also another side of biology where they're using those stem cells. Oh, interesting. Mosquitoes to do what? To take blood samples to then use for something else? And yeah, designer babies is going to be something which we can already do it at a certain extent. We can't customize babies, but what some people do is they have IVF, they create multiple embryos, they screen those embryos, and they can tell you, we can now read DNA to a certain extent, not that well. We can recognize certain traits. So simple things like hair color and eye color, they can check that before. So I can't remember the legality. I think this is like not allowed, certainly in the UK, but I'm not sure about all countries. But yeah, we're not like inserting these genes yet, but we are sort of making excess and then only implanting the ones that have the traits that we want. So yeah, it's hugely ethical, like, and open to debate, like whether this is a good thing or a bad thing. Because yeah, first, it's not available to everybody, and yeah, what traits are acceptable to screen for. So at the moment, we can screen for if the baby's gonna have a hereditary disease, so something like Huntington's disease, very severe. If you get it, like it will kill you ultimately. So we can screen embryos, and you can choose not to have the ones that have the genetic illnesses like implanted. But yeah, at the moment you can't screen for favorable traits, but this could all change in the next couple of decades. Okay, let's have a look. I saw someone asked a little while ago how I revise for GCSEs, and I wasn't like ignoring that question. There was other things to talk about, but yeah, I don't know. It was a long time ago. I can't fully remember that well. I remember doing lots of mind maps and flashcards, so I'd recommend both of those. And there's loads of apps now. You don't even have to make your own flashcards. There's loads of apps that provide flashcards to do. But mind mapping, and the most important thing is active revision. Don't just sit there reading and thinking you're taking it in. You need to be active. So write out notes, then condense them, summarize them, cover them up, try and write it from memory, like constantly be doing things, answering questions, teaching someone else, doing like questioning each other. That's active. If you're just reading and expecting it to absorb into your brain, it's not going to happen. It doesn't go in that easily. You need to be like actively doing stuff, questioning, summarizing, bullet pointing, something that's actually processing information. Then it sticks much better. If you just read it, you can remember it for about 2 minutes and then it's out of your brain again. You need to be like embedding these memories by being more proactive. Anyway, so let's look at the some eukaryotes and prokaryotes. So this is our prokaryote right down here. Oops, start it again. So they're way, way smaller. So big. Plant cells, even bigger. So prokaryotes are very small, but they can form colonies. So you can actually see prokaryotes with your naked eye. I'm sure some of you guys have used agar plates, which are like nutrient jelly. You can grow bacteria and fungi on them. And you can see, this is like, you can see a bacterial colony. So this will be many millions of bacteria all growing near each other. So they actually form a colony. Can form visible colonies. It's yeah, it's quite fun. If you guys have done this, I used to do it in my classes in school and I always enjoyed it. We got agar plates and just got people to touch it with their hand, like without washing their hands. And not like people had particularly dirty hands, but just like at a normal point in the day. And then they touch the agar plate and then you leave it for a week and see what has grown. And there's some like funky bacterial colonies you can get just off people's hands. And it's interesting. And yeah, quite often some of the people you think would be like the cleaner ones who like regularly wash their hands actually had the most bacterial colonies and fungal colonies growing from the agar. So it was interesting. And I think it shocked some people how much stuff is on their hands. It's not a bad thing. It's perfectly normal to have bacteria growing on our skin. It actually benefits our skin. So if you're overly clean, this can actually cause problems. We need these bacteria for our skin to be healthy. So completely bacterial free skin is less healthy than having some of these colonies. It's the same as in our digestive system. Bacteria actually help with our digestion. If we don't have bacteria, we don't absorb certain nutrients. Oh, nice. You swabbed your phone. Yeah, that's another good one. You get loads from a phone. Phones are disgustingly dirty. Phones and keyboards are like infinitely worse than toilet seats for amount of bacteria. So how are you guys following along so far? Nice. Perfect. So let's do an exam question. So first one, where do we find chromosomes? It is, of course, the nucleus. Where do we find prokaryotic chromosomes? Yeah, the next one is gene. Where do we find prokaryotic chromosomes? Anybody? Plasmid, nucleoid, good guess, in the cytoplasm. It's actually a trick question. They don't have chromosomes. So their DNA isn't stored in chromosomes. It's just in one big loop, which we call the nucleoloid, exactly, or nuclear coil. Coil / loop, or you can call it a nucleoloid. But they don't have chromosomes. So little trick question. Keep you on your, what's the expression? On your toes? That sounds weird, but I think that's the expression. Keep on your toes. Okay, so we've got a nice 6 marker now. We're gonna go through the question. It's a bit of a weird one, but I actually really like it. It's a good application one, so quite hard. Let's have a look. So a scientist has done an experiment with 2 types of, I actually looked up how to pronounce this just before the lesson, so I didn't get it wrong, and I've completely forgotten again. So I'm gonna give it a go. Acetabularia. No, it's not quite right, but it's whatever. We'll go with that. Acetabularia. This is a single-celled organism. So we've got 2 different types, 2 cells, each with different shaped caps. So a scientist takes parts from each cell and joins them together. So this one has got a crinkled cap. This one's got a smooth cap. The cap is like this top bit. So they take the smooth cap and they put it on to the base of a crinkled cap cell. And after several weeks, the smooth cap has become a crinkled cap. So how can we explain this? Well, there's a bit more information. So it says the shape of the cap depends on which proteins are made by the cell. Write about how making of proteins is controlled in a cell. Use this idea to explain the results and the science of the scientist's experiment. So what are we going to say for this one? Yeah, these are eukaryotic cells. So they have chromosomes. So they're cool organisms. They're actually, like, you can see them. They're the biggest single-celled organisms in existence. So you can see them with the naked eye quite easily. They're big, but they are still single-celled. So they're pretty cool. Yeah. So how come when we stuck a smooth cap onto the body of a crinkled cap, why did it become a crinkled cap? It's a bit of a tricky one. Definitely. So yeah, write about genes. Yeah, probably gonna have to write about genes. So we can say proteins coded for by genes. That's our first mark in the bank. Genes are found in the DNA sequence. Second mark, which is found in the nucleus. That's our third mark already. The cap changes shape as the cell starts to make different proteins. So the proteins that result in the shape of the cap are coming from the nucleus. So before, it was sending proteins which make a smooth cap, but now it's going to start to send proteins, it's going to send proteins that result in a crinkled cap. So over time, all these proteins that are being made, which now result in a crinkled cap, are going to mean that the cap changes to a crinkled cap. essentially. Yeah, they're single-celled eukaryotes. Exactly. So, and yeah, they actually are the biggest single-celled organisms on, at least on planet Earth. Not sure about alien life. You can't process 6 markers. Yeah, lockdown burnout. It's a real thing. Definitely. But I would just like, to be honest, I always bullet point these, and I think that's the best way to answer them. And examiners generally prefer bullet point answers. They're easier to mark. So unless it specifically says you're graded on the quality of written communication, in which case it's a bit more risky to bullet point, I would bullet point long answer questions. It helps you see where the marks are, you're awarding the marks, and helps the examiner give those marks to you. So let's put a few more points on to finish this question off. So the cap changes shape as the cell starts to make different proteins. Another mark. Yeah, you can bullet point for A-level, definitely. I highly recommend bullet pointing. Because they come from the nucleus for the crinkled cap. Crinkled. So yeah, the genes now code for crinkle cap proteins. Yeah, they're going to have alleles. So allele, you don't really need to talk about alleles in this question. An allele is like a version of a gene. So yeah, they are technically different alleles, but they also might be different genes as well. Cool. Is that question making sense? Bit of a weird one, but quite a cool question. I thought quite interesting. Everyone happy with that? From which country? I'm from England and currently in London. Okay. So I'm just looking at the time. Let's just draw a prokaryote, and then we'll finish there. We'll just draw like a sort of bog standard one. So, and label some features. Okay, I'm going to start adding some labels, and you guys can label what we're looking at. So 1, 2, that's went into this little hair. 3, 4, 5, 6, 7. What can you guys label on these? Yeah, Raheem, I was gonna talk about that one afterwards, but not draw it on because they don't all necessarily have slime capsules. I guess we can put it over here. Is 4 a plasmid? Yeah, nice answers. So cell wall, cell membrane, pili, ribosome, plasmid, DNA coil, slime coat, ribosome, and a flagella, which is the tail. So it's going to finish in a couple of minutes. Slightly overrun, basically. So we're going to end on this. No, plasmids and DNA coil aren't the same thing. Plasmids are much smaller, so they just contain a few genes, whereas the DNA coil contains all the genes, like many genes. And the plasmid is what they use to exchange. So this can go to another prokaryote. So that's the horizontal gene transfer we're talking about. We'll just quickly whiz through the functions. So cell wall is adding structural support. Cell membrane controls what goes in and out of the cell, kind of like in, both of those are like in eukaryotic cells. DNA coil stores the genetic information. The ribosome is for protein synthesis. Yeah, Elena, mesosome we could add. You don't need it for GCSE though. That's just for A level. But yeah, nice, good knowledge. Plasmid, we said, is for storing genes or transferring genes. I can't remember if I just said flagella already. Flagella is for mobility, for like moving around. So it's a bit like the tail on a sperm cell. That's like a eukaryotic version of a flagella. What else do we have? The pili. These can be used to attach to tissues or to surfaces or to stick bacteria together. So quite often they clump together to form colonies. The slime coat is a protective layer. Some have them, some don't. So it protects it. So for example, ones that live inside digestive systems quite often have slime coats to prevent them getting digested. So it's a protective layer. Or if they grow in like extreme environments, they're more likely to have it. Yeah, exactly, Zara. But again, we're not going to get into that today particularly. But yeah, good. Also good knowledge. Nice. So yeah, that's kind of all we have time for today. We've already overrun a little bit anyway. There's a couple more things I was going to do. Basically go through these a bit slower because I slightly rushed the last little bit. But yeah, hopefully you guys enjoyed it and you found it useful and got something out of it. Do you have any questions before we finish? Just checking if anyone's already asked some. Unfortunately, I don't have time to go back over those ones today, but obviously this video is going to be uploaded, so you could rewatch them and like go through it a bit slower. I'm aware that was like very rushed to explain those points. Normally I would go through it slower and we would just do it in the next class. So yeah, ribosomes are to synthesize proteins. So they're little molecules, and mRNA is translated to protein at the ribosome. So that'd be like a definition. Well, not definition, that's how it works. So videos on YouTube forever, I believe. So we do have some more sessions. So subscribe to our YouTube channel, set a reminder for these classes. So physics on Monday, biology Tuesday, maths Wednesday, chemistry Thursday. They're all at 5:30, really easy. And yeah, please join for as many as you would like to and want to come along and enter the competition. This is the link, 10 free accounts. You've got nothing to lose by signing up, so you might as well. And yeah, that's it really. Just check these questions. Yeah, you just say for bacterial, you just say that they have a loop of DNA free floating in their cytoplasm. So yeah. Reha, 3 years is the usual in the UK. So that's what I did. 3 years for a bachelor's and then another year for a master's. And nice, you still got some of the answers coming in. Nice, that's good. I can't remember what they were numbered now, so I'm presuming those are right. They look, from what I remember, they look about right. Yeah, so that's it, guys. Thanks for joining. Tune in next week. We're continuing on from this, and the other subjects. Sign up, enter the code. Hope to see you guys in the next one and on our website in a few weeks' time when we launch for good. Cool. Bye, guys. Have a good evening. Bye bye. No worries. You're very welcome. See you. Bye bye.